Hello guys, welcome to my channel. So as you know, we are studying about the carbohydrates. So we did monosaccharide, we did disaccharide. Now we'll move to polysaccharide. Okay, so we'll just put here that we are doing polysaccharide because this might be useful for us. Polysaccharide, right? So we're studying about the polysaccharides. So as we know, most carbohydrates found in nature occurs as a polysaccharide. It's not like as a monosaccharide or a disaccharide, but like in nature, it's actually a polysaccharide. That is, what is polysaccharides? That is polymers of medium to high molecular weights connected as a monosaccharide, right? So polysaccharides also called glycans. What are they? Are called also called glycans, right? So they are also called glycans. So the polysaccharides differ from each other in identity of their recurring monosaccharide units. So how the polysaccharides differ to each other as of recurring monosaccharide units in the length of their chain or in the types of bonds linking the units and the degree of branching. So this is how they are differ. So it differs in the branching, it differs in the linkings of the unit, that is the bonding, and also different monosaccharide units. So for example, there are two types of the polysaccharide. So one is homopolysaccharide, right? So the one is homopolysaccharide and one is heteropolysaccharide. All right, so these are the two types of polysaccharides. So these are saccharides and not just one. So homopolysaccharides. So what do you mean by homopolysaccharide? That means containing only a single monomeric species, right? So it has a single monomeric species. For example, if it says glucose, so the entire chain will be of a glucose. But for the heteropolysaccharide, it can be glucose, it can be other different protein or anything else. So that is a heteropolysaccharide, right? That contains the hydropolysaccharide that contains two or more different kinds, right? So the some homopolysaccharide serves as a storage. So what are the function of homopolysaccharides? So some serve as a storage form of monosaccharides that are used as a fuel. So this, right, the homopolysaccharides serves as a storage form of monosaccharide, storage form of monosaccharides. So they serve as a storage forms of monosaccharide. That monosaccharide are used as a fuels that, for example, that are like the example are starch and glycogen, right? Right. So these are the example of homopolysaccharides, right? So there are other types, but like beside this, there are other types of homopolysaccharides as well, like such as cellulose and cytine, like other types as well that are cellulose and chitin. So this what are the function of these two? They serve as a structural element in the plant cell walls and animal exoskeleton. So these are the different other types of homopolysaccharide, and stru- that serves as a structural element in plant cell walls and animal exoskeleton. All right. So these are the example of homopolysaccharides. So what are the example of heteropolysaccharides? So the heteropolysaccharide, what's the function? It provides extracellular support for the organisms of all kingdoms. Right. So the function of heteropolysaccharide, what it does, it provide an extracellular support. Right. Extracellular It provides extracellular support for the organisms of all the kingdoms. Organisms of all kingdoms, right? So that is a function of heteropolysaccharide. Like for example, the rigid layer of the heteropolysaccharide, right, that envelopes the cell, that is composed of heteropolysaccharide built from two alternating monopolysaccharide, right? So there will be like two alternating monosaccharide that is built a long chain that composed the heteropolysaccharide, that is peptidoglycan, for example. So in animal tissue, for example, the extracellular space is occupied by several types of heteropolysaccharide, right? So like the in animal tissues, right, the extracellular space is occupied by several types of heteropolysaccharide, which form a matrix that hold the individual cells together and provide protection, shape, and support to cells, tissues, and organs. So these are the functions of heteropolysaccharide. What it does, it exists in an extracellular space, right? And what it does, it forms a matrix. And what the matrix do, it holds the individual cells together, provide protection, shape, support to the cells, tissues, and organs. Right, and this homopolysaccharide and heteropolysaccharide have like a branch, unbranched structure and a branch structure. Like for example, this is just a simple homopolysaccharide or an like homopolysaccharide, for example. This has like same 000, for example, and this is unbranched. It does not have any unbranched, right? This is unbranched, and like for example, branches is like this is also homo polysaccharide, and it has like for example this here it goes, it has a branch. So this is a, for example, a branched homopolysaccharide. And for example hetero, so like hetero is has a different kinds, right? So it might be like star here, and like maybe some this kind, some square here, whatever. So this is the heteropolysaccharide. It has a different monosaccharides unit at some point. So this is unbranched, and if had like something like here, right? So this is a branch. So these are a type of a homopolysaccharide and heteropolysaccharide. Right, and the polysaccharide, like unlike proteins, generally do not have any defining molecular weights. Like for example, proteins have a defined molecular weight, but like the polysaccharide does not have any defined molecular weight. Why? Because the difference is a consequence mechanisms of assembly of the two types of polymers. So for proteins, they are synthesized on the template, right? That is a messenger RNA. They are synthesized on a messenger RNA. So that has a defined sequence, length, and by enzyme that follow the template exactly. So that's how the protein is has a defined molecular weight, because it is exactly the same. But for polysaccharide synthesis, there is no template. It does not have any kind of template. Rather, the programming for the polysaccharide synthesis is intrinsic to the enzyme that catalyzes the polymerization of the monomeric units. So there is no specific stopping point in the synthesis process, and the product does vary in the length. So some is like very long, some are short. That's why it does not have any defined molecular weight. Right. So now the some homopolysaccharides are the storage forms of fuels that we studied here, the starch and glycogen. So the most important storage polysaccharides that are homopolysaccharides are the starch in the plant cell. So the starch in the plant cell and glycogen in the animal cell. And this both polysaccharides occur intracellularly as a large cluster or granules. So this occurs intracellularly. How they are seen in the intracellular, it's like as a structure of clusters or granules. And starch and glycogen molecules are heavily hydrated. Why they are heavily hydrated? Because they have many exposed hydroxyl groups available to hydrogen bonds with water. Like, right, you can see these are hydroxyl groups that are available to bond with water. That is a hydrogen bonding with the water. So most plant cells have the ability to form starch, right? And the starch storage is especially abundant in tubers, that is under the stem, right? Such as potatoes and seeds, right? So they can store the starch. So the starch contain two types of glucose polymers. So starch contain two types of glucose polymers. Sorry. So write down is contain two type of glucose polymers. That is number A, amylose, right? And B is amylopectin. Right, so these contain two types of glucose polymer, that is amylose and amylopectin. So the amylose, the first one, consists of a long unbranched chain. So this is unbranched chain of D-glucose, that is the D-glucose residues connected by, so they are connected by, so this contain unbranched chain of D-glucose connected by alpha 1 to 4. Remember the alpha 1 to 4 linkage? This is how it's connected. So alpha 1, so this is the alpha first glucose, that's why I did not remove this. So alpha 1 glucose, and this is connected by, and this is the number 1 carbon atom here, and here is the number 4 carbon atom here. So that's why it's alpha 1 to 4, right? So the amylose consists of a long and unbranched chain. So these are not unbranched chain of this D-glucose residues connected by this alpha 1 to 4 carbon linkage, as in maltose, right? Remember when this two connects and it forms maltose, correct? So such chains vary in the molecular weight from few thousands to more than a million, right? So the molecular weight can be few thousands to more than a million. But for amylopectin, it also has a higher molecular weight, that is up to 200 million for amylopectin. But like amylose is highly branched. So this is unbranched and this amylo is highly branched chain. Amylopectin, sorry, is highly branched chain, right? So the glycosidic linkage joining successive glucose residue in amylopectin chains are alpha 1. So as this is a branch one, right? So the glycosidic bonds or the residues, glycosidic linkages joining the glucose residues joining together. So the amylopectin chain has this, of course, alpha 1 to 4, this linkage, but the branch points. So the branch points is alpha 1 to 6, right? So this is alpha 1 to 6. So this is joining the branch point. So like for example, this is the branch point. So it is joining the alpha 1, that's the first carbon atom, and the 6th carbon atom on the chain, that is the 6 is here. So it's like this joining here on the branch point, right? And this occurs every 24 to 30 residues. The branching occurs every 24 to 30 residue, right? So like 1, 2, 3, 4, 5, and like when it's 24 to 30, it branches off, right? And these are the linkage. All right, so this was starch. Now glycogen, right? So this was storage for plants and this is storage for animals, right? This is the main storage polysaccharide of animal cells. So it has also the same thing, but like amylopectin, glycogen is a polymer, the same alpha 1 to 4 glucose subunit. It branch the same alpha 1 to 6, but the glycogen is more extremely branched. So this is 24 to 30, but the glycogen branches every 8 to 12 residues. So this branches 24 to 30 residues, but like this branches very frequently 8 to 12 residues, and these are more compact than starch. Glycogen is especially abundant in the liver. Remember, so the glycogen especially abundant in the liver. All right, where it, and in the liver it may constitute as much as 7% of the wet weight, and it is also present in the skeletal muscles, right? So especially abundant in the liver and also present in skeletal muscles, right? So, as in it's in the liver. So, in hepatocytes, that is in liver, glycogen is found in large granules. So, how glycogen is found in large granules? The starch is found in the clusters or granules like starch and glycogen. So, but the glycogen in the liver is found in large granules, which are the clusters of smaller granules composed of single highly branched glycogen molecules with an average molecular weight of several millions. So when you can see it's large granules, but the large granules are the clusters of smaller granules that are composed of single highly branched glycogen molecules with an average molecular weights of several millions. So the large glycogen granules also contains in tightly bound form the enzyme responsible for synthesis and degradation of glycogen. So the large glycogen granule, so the large glycogen granules also contains the enzyme that is responsible for the synthesis and the degradation of the glycogen, right? Yeah, so because each branch in the glycogen ends with a non-reducing sugar unit, right? So as you remember when we discussed disaccharide, so when two disaccharides join and when there is an anomeric and free anomeric carbon at the end, so it is a reducing sugar, but if it does not have, then it's non-reducing. So in the glycogen with a non-reducing sugar unit, it has a non-reducing sugar unit where it's along the branch. A glycogen molecule with n branches has n plus 1 non-reducing ends, right? But only one reducing end. So it has number of non-reducing end but just one reducing end. So when the glycogen is used as an energy source, glucose units are removed one at a time from the non-reducing end. So for example, this is a glycogen, right? And these are all the non-reducing end, it's just one reducing end. So when the degradation start, when the glucose units are removed one at a time, so these all glucose units are removed at one at a time, that are removed from the non-reducing ends, and the degradative enzyme, as we know, it's inside, that act only at the non-reducing ends, can work simultaneously on many branches, speeding the conversion of the polymers to monosaccharide, right? So when this glycogen is used as a energy source, this is degraded by the enzyme and like it's separated in all the monosaccharide. So the question is why not store glucose in its monomeric form, just like this, right? If it's you at the end it's separated for the energy, it's not stored separately, because if you calculate, it has been calculated that hepatocytes store glycogen equivalent to a glucose concentration of 0.4 moles, right? So the actual concentration of glycogen, which is the glycogen is insoluble and contributes little to the osmolarity of the cytosol, it's about the actual concentration is 0.01 micromole, right? So if the cytosol contain 0.4 mole glucose, the osmolarity, so if it's stored separately, imagine, so the cytosol will contain this amount of concentration of glucose, so that means the osmolarity would be threateningly elevated, and if the osmolarity is elevated, that leads to the osmotic entry of the water inside the cell, and that might rupture the cell, and that is the reason glucose is not stored as a monomeric unit in the hepatocyte, but it's stored as a glycogen, right? And one more thing, beside this, the intracellular glucose concentration is 0.4, and an external concentration of glucose is about 5 micromoles. So that means the concentration in blood of a mammal. This is concentration in blood of a mammal. So free energy change of the glucose uptake into the cell against this very high concentration gradients would be prohibitively large. Right? So these are the reason the glucose is not stored monomeric. Right? Beside this starch glycogen, there are dextrins. So there are dextrins. So these dextrins are bacterial and yeast polysaccharides. So what are they? They are bacterial and yeast polysaccharides. Bacterial and yeast polysaccharides. These are made up of alpha 1 to 6 linked. So these are made up of this link poly D glucose, and all have alpha, instead of alpha 1 to 4, it has alpha 1 to 3 branches, and some also have alpha 1 to 2 or alpha 1 to 4 branches as well. But the dextrins, that is dental plaque formed by bacteria growing on the surface of teeth, is rich in dextrins, right? So the dental plaque right, that are formed by bacteria on our teeth, on the surface of the teeth, these are rich in dextrins, which are adhesive and allow the bacteria to stick to the teeth and to each other. So dextrins also provide a source of glucose for a bacterial metabolism, right? Synthetic dextrins are components of several commercial products. There are synthetic dextrins as well, and those are the components of several commercial products. For example, Cefadex, that is used in the fractionation of proteins by size exclusion from the chromatography. The dextrins in this product are chemically cross-linked to form insoluble materials of various sizes. Alright guys, so this was homopolysaccharides. So there are some other homopolysaccharides as well that we discuss, cellulose and cytine. So we will discuss in the next video. I think this is enough for this starch, glycogen and dextrins. Alright guys, so we'll see you in the next video. We'll continue with the homopolysaccharide and we'll move further with the cellulose and cytine. Alright, thank you guys for watching and don't forget to subscribe to the channel.